Okay, so we're going to talk about how we get energy from lipids. So, about 1/3 of our energy comes from dietary triacylglycerols. So remember, the triacylglycerol is the molecule that we saw. It looks like this. Obviously, this is just a cartoon. This would be a glycerol molecule, and it's a triacyl. So it's going to have 3 of these fatty acid chains, and it could have a double bond and be saturated or unsaturated. So we get these triacylglycerols from our diets, and we use them as stored energy. And some of our organs, like our heart and our liver, they prefer fatty acids and triacylglycerols as their primary energy source. So, as we look at what we learned about cellular respiration, actually on slide number 41 of this lipids chapter, it shows how these parts of a triacylglycerol go into what we have already learned about cellular respiration. So the glycerol molecule, once these bonds are broken and these fatty acid chains are cleaved off of this glycerol molecule, glycerol is going to get converted into glyceraldehyde 3-phosphate. And from there, it's going to get fed through the rest of glycolysis. Remember, glyceraldehyde 3-phosphate is one of the intermediates for glycolysis. The rest of these fatty acids, they are going to get converted into acetyl-CoA. So remember acetyl-CoA, these are 2 carbon pieces. Because remember, we make a pyruvate, which is a 3-carbon piece, but then we lose a carbon dioxide, which removes a carbon when we go from pyruvate to acetyl-CoA, and then acetyl-CoA goes into the citric acid cycle. So the glycerol from the triacylglycerol, or the triglyceride, is going to get converted into glyceraldehyde 3-phosphate and go through glycolysis. The fatty acids are going to get broke apart into 2 carbon pieces. Each of those 2 carbon pieces will be converted into acetyl-CoA, and then acetyl-CoA goes into the citric acid cycle, and then it continues through the rest of cellular respiration that we've already learned about. The next few slides on page 42 and 43 are kind of a review of what we talked about with the integration of metabolism, showing what type of fuel different organs prefer. And we'll come back to this again, but remembering that our heart, our brain prefers glucose. It can use ketones, which we'll talk about at the end of this. But there are a few organs that prefer fatty acids as their major source of fuel. Okay, so fatty acids actually can carry the most energy per carbon because they're the most reduced. And they are anhydrous because they're nonpolar. So anhydrous means without water. So they have more energy per gram. So this is not a nutrition class, but if it were, we would learn that fats have about 9 calories per gram, carbs have about 4 calories per gram, and proteins also have about 4 calories per gram. So just if you're looking at the amount of energy per mass, fats have the most energy per mass. And the reason for this is that they're anhydrous and they're the most oxidized, so they're able to carry the most energy per carbon. So we learned about glucose and glycogen, and glucose and glycogen are short-term energy needs. So they deliver quick energy. Also, they're very hydrated, so they give us less energy per carbon. Fats are for long-term energy needs, and they have a slow delivery. So fats are digested in our digestive system, and then they're broke apart by the different enzymes and bile salts in our digestive system. And they can either be stored or they can be utilized for energy through a process that is called beta oxidation of fatty acids. So I put some slides in our lecture notes from a few different textbooks, so you're going to see a little bit of a review. The first thing that I wanted to go over was this step of how glycerol becomes glyceraldehyde 3-phosphate. So this process here is actually on slide number 57, where it shows the conversion of glycerol to glyceraldehyde 3-phosphate. So these enzymes, this actually happens in the cytoplasm, where glycerol is converted to glyceraldehyde 3-phosphate, because remember, glyceraldehyde 3-phosphate going into glycolysis also happens in the cytoplasm. This next part, I'll put that here, cytoplasm. The fatty acid oxidation happens in the mitochondria. So one thing that's important is that these carbon pieces, these long carbon chains, if the carbon chain is small, so what small means is that it is less than 12 carbons, that means it can diffuse freely across the mitochondrial membrane. If they're large, they have to use what's called a carnitine shuttle. Sometimes it's called an acylcarnitine transporter. To get into the mitochondria. So smaller carbon chains can diffuse freely into the mitochondria. Larger carbon chains need some help. They need this carnitine shuttle. And this carnitine shuttle, a good picture of that can be found on slide number 60. So this carnitine is this transport protein that attaches to the larger carbon chain, and it goes through this transport protein on the inner mitochondrial membrane, and it transfers it into the mitochondrial matrix. Remember, ultimately, these acetyl-CoA pieces that we're going to break off of this fatty acid are going to go through the citric acid cycle. And remember where the citric acid cycle happens. It happens in the mitochondrial matrix. So it's important to pay attention to where these cellular processes are happening. Okay. So there's 3 major stages of fatty acid oxidation. And sometimes you'll see this called beta oxidation of fatty acids. Same thing. And I used a few slides from a few different textbooks or different sources. So if you see it kind of used interchangeably, that's why. And it means the same thing. Okay, so the first step is the conversion of the 2 carbon units that have been cleaved off into acetyl-CoA. And that's going to make NADH. The next step is going to be the oxidation of acetyl-CoA into carbon dioxide and the citric acid cycle. In the citric acid cycle, it's going to produce GTP, NADH, and FADH2. So acetyl-CoA into the citric acid cycle. And then the third step is going to be oxidative phosphorylation. On the electron transport chain of all of the intermediates that are made on the citric acid cycle. So really only step 1 is different. Step 1 is converting these 2 carbon pieces into acetyl-CoA. And then from step 2 and 3, it's just like we were in cellular respiration where we started with glycolysis. Okay, so this is called beta oxidation because it's cut off at the beta carbon. So, for example, where this glycerol is, once this glycerol has been removed, we have the beginning of the chain. So let's say here is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I drew it too long. Say it ends here. So this would be C12. This is a 12 carbon chain. So what happens is, if this is the first carbon, this right here would be the alpha carbon, this right here would be the beta carbon, and then it goes all the way down. So this piece right here would be cleaved, and now this 2 carbon piece is going to go off to be converted into acetyl-CoA. A CoA would be added, and then it would go on. Now this is the alpha carbon and this is the beta carbon, so then it would cleave here, and then this piece would go off to make an acetyl-CoA. So if you have a 12 carbon fatty acid chain, you could make 6 acetyl-CoAs. So when you do all of this, there's a sheet at the end that shows kind of the balance sheet of how much energy we make in a fatty acid versus glucose. So for example, ATP during the oxidation of 1 molecule of palmitoyl-CoA. So palmitoyl-CoA, I think palmitoyl is, I'm not 100% sure. I think palmitoyl is 16 carbons, but for each of the palmitoyl, no, sorry, it's 14 carbons. So for that, you would make 108 ATP for 1 palmitoyl-CoA, compared to 1 molecule of glucose only making 32 ATP. So fats do store the most energy because they're anhydrous and because they are the most oxidized, and they're going to produce the most energy. So fatty acid oxidation will only happen if there's no other fuel immediately available. And this is the whole premise behind a low-carb diet. If people are trying to lose fat, stored fat in our adipocytes, our adipose tissue, or fat in our diet, we will not use that fat as a primary source of fuel. We will always use glucose and glycogen as a primary source of fuel first. That's what's the most energetically favorable. So the premise behind a low-carb diet is, if you're not eating a lot of carbohydrates, you don't have a lot of stored glycogen, then you will burn stored fat, because you'll only ever burn stored fat if there's no glucose or glycogen around. Another process that we're going to talk about is something called ketosis and the formation of ketone bodies. Okay, so the premise behind this, so ketosis, or a ketogenic diet, is also kind of a fad right now. And what a ketogenic diet does is it puts you in ketosis, where your body is burning ketones. So let's go back a little bit. So if we remember the citric acid cycle, So remember, what comes into the citric acid cycle is acetyl-CoA. And the first intermediate here is citrate, and then it goes around and around. Well, the last intermediate over here is called oxaloacetate. And oxaloacetate is a really important molecule. It's a really important regulator of which of these metabolic processes are active or inactive. You need oxaloacetate for the citric acid cycle to continue. It doesn't matter how much acetyl-CoA you have to bring into the citric acid cycle. If you don't have oxaloacetate to combine with the acetyl-CoA that's coming in, you cannot form citrate. So low oxaloacetate will stop the citric acid cycle. It'll stop the TCA cycle. When the TCA cycle is stopped, acetyl-CoA will be converted into something called ketone bodies. The first step of forming ketone bodies is joining two acetyl-CoA subunits. So we would take two of these acetyl-CoA subunits that we had broken apart from fatty acid oxidation and form a ketone body. So the production of ketone bodies increases during starvation, where we're using mainly stored fat for fuel. This happens in our liver, and our organs can use ketone bodies as fuel, especially our brain. One of the problems with ketosis is one of the byproducts of ketogenesis is very acidic and can make your blood pH too low. And this is sometimes a process, sometimes a byproduct of people with diabetes, and when their blood sugar isn't regulated, if they don't have any blood sugar around and they're making ketone bodies, those ketone bodies that they're making, one of them is actually acetone. So you might smell someone's breath, smell like acetone, or almost smell a little bit sweet, and it could be from this process, from the ketosis that is being unregulated. Okay, so that's ketosis. So fats are an important energy source in animals. These 2 carbon units from our fatty acid chains are oxidized by beta oxidation. It's broken apart into 2 carbon pieces until all of these carbons are used up. Remember that natural fatty acids are made in even numbers, so you would always have an even number here. We're not going to talk about synthetic fats if you had an odd number. And then these acetyl-CoAs just go right into the citric acid cycle, unless it's a time of starvation when there's no oxaloacetate around. Then your body could take 2 of those acetyl-CoA pieces and make ketones that then serve as fuel when there's no other types of fuel around. So this is one more process that we can put into our integration of metabolism, and kind of look at a bigger picture of which processes are going to be active or inactive in various cellular situations.